Good morning, everyone. Thank you for listening to our work. So today I'm going to briefly describe our work in the last 10 years about mechanisms of genome instability, basically genome damage and repair defects and their consequences in brain diseases, and whether these mechanisms or pathways which we and others have identified that have emerged in the last few years can be potential therapeutic targets or adjunct targets for ameliorating brain disorders. As you know, briefly in a couple of sentences, as you know, genome damage has been linked to various diseases, most of the diseases, and particularly brain diseases, including neurodegeneration, stroke, and even brain injury, and so on. But genome damage or repair targeted therapies have not been, explored in the clinics or have not been well developed. One of the reasons is um, the initial studies about a decade back, most researchers thought that genome damage is a consequence, various pathways lead to genome damage, and it's just a consequence, it's one of the byproducts of neurodegeneration. But now we know that whether it may or may not be the initial cause of the disease, but genome instability significantly contributes to the disease progression or the severity of the disease and the duration of the disease and so on. And targeting DNA repair may be a great option to slow down the neurodegeneration as well as to minimize the symptoms and increase the longevity. So before I start, I want to acknowledge the people in my lab who contributed to the work, and most of the work I am presenting is done by these people. Primarily today, I'm going to present the work done by Haibo Wang, who is a junior faculty in our lab who works on first and ALS, and Joy Mitra, who has been recently promoted as instructor. So, and there are other people, both current members and former members here. Manohar Kodavati, Vikas Rao, and Vincent Prosek and Pavana Dikshit. They all contributed to the work in one way or the other, and some of the former members as well. So I would also like to acknowledge the current fundings in our lab, apart from NIH grants on ALS and stroke. We also have a industry funded grant from a drug discovery company to screen or to develop DNA RNA oligonucleotide based therapeutics for ALS, and also the Houston Methodist for various support. So as Dr. Singh mentioned, research in our laboratory is focused on understanding the mechanisms, molecular mechanisms of genome damage and repair responses, as well as its consequences in neurons and other cell types in the central nervous system. So in some cases, you can call it as, we already know that damage occurs or, this repair pathway is affected. In some cases, it's like revisiting those pathways to understand the, key molecular pathways or key rate limiting step, which can be therapeutically targeted. And also to understand what is the consequence in neurons, what happens when those particular DNA damage happens, what is the consequences in neurons, whether it leads to cell death, whether it leads to senescence, or, what is the immediate or long-term effect on neurons. And major focus in our lab is on role of RNA DNA binding proteins like TDP43 and FUS in motor neuron disease, as well as other dementia-associated neurodegenerative diseases. And our lab specializes in using human iPSC-derived neuronal models, including several patient-derived neurons who have had familial ALS with specific mutations in these RNA DNA binding proteins. And also, we use gene editing methods to generate isogenic cell lines or making various pathological modifications. And we have some relevant mice models, rodent models of these neurodegenerative diseases, including some of them which we generated in our lab, which I'm going to briefly mention, and also human disease specimens. So as I mentioned, although we knew that DNA damage has been intrinsic to these diseases, the major roadblock was we did not know the specific understanding of the mechanism or specific pathways or the specific repair component affected in particular disease subtype. That was one of the roadblock for exploring DNA repair targeted interventions. And my studies, which I'm going to present now, will shed some light on these aspects. And we will also know one of the challenging questions people have been discussing for a long time: how and why neurons are particularly affected for this DNA repair defects. And I would like to, from our studies, we at least we'll answer this question at least partially with one of the mechanisms. And the today I'm going to, because of the time constraint, limit my presentation to the three important works which we published in the last 2 years. One about first time identification of TDP43, an RNA binding proteins involved in about 95% of all motor neuron disease and about maybe 50% of dementia associated diseases. Its role in DNA double strand break repair, which we published a couple of years back. And another related RNA binding protein, which is mostly involved in familial neurodegeneration or familial motor neuron disease, which is FUS. And it plays a role in a different DNA repair pathway. And in rare cases, both these protein pathologies can occur, and that kind of compounds the issue of the DNA damage contribution. And a third paper on stroke or hemorrhagic stroke, where we identified blood degradation products contributes to long-term or chronic neuronal dysfunction via its DNA damage role, as well as damage-induced senescence-like state in neurons. And we also developed, based on as a proof of concept, we are applying the same strategy for other cases. So as a proof of concept, based on this mechanism, that mechanistic derived combinatorial therapy using a nanoparticle, which is both antioxidant and a metal chelator, which has been very effective. So this has been published in ACS Nano. So as you all know, there are more than at least 200 types of neurodegenerative diseases known in humans, and most of all of them are progressive and age associated, and also 90% of them are sporadic, and about 5 to 10%, depending on the disease subtype, could be familial with inherited mutations in a particular protein. And if you look at the etiology, they seem to have common etiological factors, metal toxicity and oxidative stress, protein misfolding and aggregation like amyloid, tau, synuclein, and Parkinson's, and more recently, RNA DNA binding proteins like TDP43 and FUS in amyotrophic lateral sclerosis. But the third common feature is accumulation of DNA damage, which was kind of, I don't say that it's ignored, but people thought it's kind of, it happens, without much attention to it. But one of the things which we don't know is the crosstalk among these three common etiological factors, which occurs first and what is the interaction among them for the, impact and their impact on disease progression and so on. And one important aspect, briefly before going to our studies in a couple of slides, one important aspect or you can call feature or a challenge of neurons is, we have most of our neurons in the adult body which have developed within 3 years of life. And brain is highly metabolically active even when we are sleeping. Its transcriptional activity is high. So it consumes a large amount of oxygen compared to other organs in the body. So genomes in the neuronal cells or brain cells are continuously exposed to various redox environment and damage. So accurate or efficient repair of those damage is extremely important. And because we develop these neurons within 3 years of age, and then they live as long as we live, they have a long lifespan, which makes it the impact of environment in accumulating some chronic changes becomes extremely important. So I already mentioned that the oxygen and redox environment is high in neurons, and they are always susceptible to various types of DNA damage. And these are some of the common DNA damages. Oxidized bases, slightly modified bases like methyl and alkaline bases, and single strand breaks, double strand breaks, cross links, and so on. So even just oxidized bases, there could be several dozen oxidative modifications. So these occur continuously, like as per one estimate, about 10 to the power of 6 base damages occur in an aerobic cell per day. And if this level of damages occur and we still survive, cells are functioning over a long period of time, that means we have developed multiple repair pathways or multiple systems to repair or fix these damages. And that's exactly what mammalian cells or higher organisms have developed. And in fact, for each damage, you have multiple repair pathways. For example, for double strand breaks, which are the most lethal form of damage, you have three different repair pathways: homologous recombination, which occurs in dividing cells, and non-homologous unjoining, which occurs in most cell types, and an alternative unjoining, where single strand break repair machinery is co-opted for repair pathways. And about about 5 to 10 years back, we were looking at complexity of mammalian DNA repair, and we found that you need about 4 to 5 key enzymes to repair a oxidatory damage or single strand break, but there are about 3 dozen different non-canonical factors involved. We published several papers, series of papers on the role of these non-canonical factors in higher organisms. And one key you know non-canonical factor turned out to be RNA binding protein. Like we found HNR and PU playing a role in oxidative damage repair, especially when the damage is induced. And it also kind of, when there are complex damages, HNR and PU acts as a molecular switch to kind of hierarchically repair the damage, to so that a most lethal form of damage is repaired first, followed by other damages. Around the same time, other group of RNA binding, DNA binding proteins like TDP43 and FUS were implicated in motor neuron disease, initially by Virginia Lee in 2006, and subsequently by several other groups. And Paul Taylor in St. Jude's in 2010 was looking at TDP43's interacting partners to identify its potential role in neurons. And one of the proteins in that proteomic study was COO. And because we were looking at HNR and PU and their DNA damage role, and TDP43 associates with the DNA repair protein, then I got interested into it, and we started looking into TDP43 and FUS's role in DNA damage response. And I should also mention that ever since TDP43 was implicated in ALS, subsequent studies show that about 40 to 50% of all neurodegenerative diseases at some stage of the disease progression have some kind of RNA DNA binding pathology. And ALS particularly, I would like to specifically mention this, compared to other neurodegenerative diseases, ALS is unique. Like you have at least more than a dozen factors involved in these diseases. And in many cases, these are unique, they don't overlap. In few cases, you may have TDP43 and C9orf overlapping, or TDP43 and first overlapping, maybe in 1 to 5% cases, but most of the time they are unique. That means although the symptoms are like motor neurons disease symptoms, so underlying pathology is distinct. There could be like 10 to 15 different disease subtypes. So, a symptomatic improvement is different thing. But if you have to develop therapy, you need to first understand what underlying pathology exists and what is their mechanism of action, pathophysiological action. And this was a challenge. So, we started looking into TDP43's role in DNA repair. And following up on Paul Taylor's study, we found that TDP43 in fact associates with most of the DNA double-strand break repair protein, and this is in damage-dependent manner. Like if you induce damage by radiation or etoposide, you can see the interaction increases by several folds. And this was done by human-induced pluripotent cell-derived motor neurons. And this is a proximity ligation assay. For those of you who don't know this is very specific for cell in situ protein interaction. When you have two proteins in close proximity, you can look into video. We use two antibodies to those proteins of interest, and the secondary antibodies have small DNA oligos with complementary sequences. So if proteins of interest are in close proximity or in a complex, these oligos can anneal together to form a circle, which can then be amplified by rolling circle mechanism. And then you can use a fluorophore tag to a complementary oligo and to detect that interaction. One foci or one spot indicates one interaction. And these interactions were specific. You can see that TDP43 interacts only with double-strand break associated with associated ligase, XRC4 ligase 4 complex, but not single-strand break repair associated XRC1 and ligase 3. And then we looked at whether TDP43 goes to the DNA double-strand breaks. If you can see this in this video, we tagged TDP43 with a fluorescence dye like GFP, and as soon as you induce laser damage, TDP43 goes on the laser track. And this is as fast as the classical DNA double-strand break repair protein COO. And in fact, TDP43 also follows similar dissociation kinetics as Ku. And we then looked at the impact of loss of TDP43. We initially looked at siRNA mediated knockdown or shRNA mediated knockdown. I am showing the data of CRISPR knockout here. When we knocked out TDP43 by CRISPR, cells started dying within 2 to 3 days because it's an essential protein. So to overcome this, we made a conditional CRISPR cell line where you can gradually knock down TDP43 by doxycycline treatment, and around 6 days cells start dying, 6 to 8 days. So you have at least 2 to 4 days where you have gradual decrease in TDP43, and this resulted in significant increase in DNA double strand breaks. So you can see that that's a dose dependent increase in double strand breaks as you knock down TDP43, and these are mostly double strand breaks. As you can see here, a comparison of alkaline and neutral cometastasis shows similar level of damage, which suggests that most of the damages are double strand breaks. So then we looked at the impact of TDP43 on double strand break repair itself using a surrogate plasmid-based NHEJ assay. You can see here in control cells, this is the amount now, level of repair. If you knock out TDP43, the NHEJ efficiency goes down significantly. We did a series of experiments, which I'm not going into, individual details, but overall, we found that TDP43 plays a role in double strand break repair via NHEJ, and we found a similar correlation in human brain tissue where there is TDP43 pathology, which is a nuclear clearance. TDP43 is predominantly nuclear protein. Under diseased condition, it sequesters in the cytosol, primarily in stress granules and so on. So it will have like loss of nuclear TDP43 and its gain of toxicity in the cytosol. And you can see in human tissue that correlates with strong tunnel staining as well as gamma 2X staining, which is a DNA damage marker. And also by Western, we found that compared to control, you have huge increase in DNA damage markers. And finally, we wanted to understand the mechanisms. Again, very briefly, one piece of data I would like to mention. So what exactly TDP43 does in double strand breaks? So we found that TDP43 is involved in recruitment of the final reaction enzyme for double strand breaks, which is XRCC4 ligase 4. So if you don't have TDP43, DNA double strand break ligation is affected. And we found that in fact, TDP43 is required for stabilizing the ligase complex at DNA double strand breaks. And as I mentioned, TDP43 has dual role. One is like its nuclear role, as well as in the pathology, it has a cytoplasmic aggregation. To understand whether the DNA damage is because of its loss of function or gain of toxicity, we specifically knocked out TDP43 from neuronal nucleus by mutating its nuclear localization signal, and found that its nuclear specific loss leads to DNA damage, not the cytoplasmic specific loss. And this was important for us to mimic the disease pathology, because this is what happens in the disease. You don't get overall TDP43 knockdown, but you see like only nuclear knockdown, but TDP43 accumulates in the cytosol. So to take it further, we developed a mouse model based on this. What we did was we developed a conditional mouse model where you can specifically knock out TDP43 in the nucleus and increase nucleocytoplasmic translocation. So what we did was we added a mutant nuclear NLS containing exon in the next intron of E3, exon 3, which is the NLS containing exon. And we put a combination of wild type and mutant LOXP sites. So when we cross with CRE, the wild type exon containing NLS is deleted, while the mutant NLS comes in the right orientation and it expresses, and you can get the disease specific nuclear clearance. And this actually, I'm showing the DNA damage data. It accumulates the DNA damage as well as motor functions and behavioral defects. And we did a lot of studies with this mice model. And the main goal was to use it for therapeutic screening, because we wanted to mimic human pathology, not just TDP43 knockdown or transgenic overexpression and so on. And the advantage of this mouse model is because it's conditional, we can now use a neuron specific CRE or astrocyte specific CRE or central nervous specific CRE. We can knock out in cell specific manner and look at the impact. And that's exactly what we are studying. So I mentioned that TDP43 is required for recruitment of XRCC4 ligase 4 complex. When you have a TDP43 mutation which has more cytoplasmic presence, so we found that traps XRCC4 in the cytosol and you can see it's excessive for ligase 4 is trapped in the cytosol. So it prevents this complex going to nucleus when there is DNA damage. So it basically prevents the recruitment of excessive for ligase 4 into the nucleus and onto the chromatin. So in summary, we show that TDP43 is required for DNA double-strand break repair, and the persistent DNA double-strand breaks, which accumulates under TDP43 pathology condition, contributes to neuronal inflammation, neuronal senescence-like state, and motor neuron disease progression. We published a series of papers here, including the PNAs, which first time showed that role in double-strand breaks. And very briefly, I think I will have to wind up in about 5 minutes. I mentioned a related protein first plays a role in double strand in DNA strand break repair, not in double strand breaks. It's very interesting. This is involved in familial ALS, and Haibo Wang published a Nature Communication paper where he showed that it plays a role in oxidizer base repair, unlike TDP43. So this actually associates with DNA ligase 3 XRCC1 complex, and it stabilizes that complex. And here we did systematically, like we had, because this is involved in familial ALS, we had patient-derived cells with first mutation, and we found ligase 3 defect in two of the patient-derived cells with different mutations. So if you correct that mutation by CRISPR-Cas9 knock-in, we could get the ligase 3 activity back. This shows that this ligation defect can be attributed to first mutation. And the ligase 3 defect is in fact because of, you can see the recruitment of XSAC1 ligase 3 complex to the damaged site. If you have first wild type, the XSAC1 ligase 3 goes to the lesser damaged tract immediately. And in the mutant case, it not only delayed recruitment, but intensity is much low. And we did multiple complementary approaches to show that. And there are two additional dynamics on this. This is important. I'll very briefly mention. We also found that first actually regulates PARP1's function, which is important for DNA repair as well as stress granule formation. So it kind of has a dual role, like kind of double whammy kind of function, where it's required for nuclear function, but it is toxic in the stress granule formation. So we are trying to approach a combinatorial strategy where you take care of ligation complementation for relieving the nuclear DNA damage and targeting PARP1 in the cytosol to prevent its toxicity in the stress granules. And we have very promising results in the cultured cells. We are taking it further. And another important thing is in nucleus, there are three ligases, ligase 3, ligase 1, and ligase 4. But in mitochondria, there is only one ligase, ligase 3. So we found that because of that, the impact of first is much more in mitochondria. We found that it actually leads to mitochondrial genome instability and mitochondrial oxidative stress and so on. And overexpressing a ligase in mitochondria actually ameliorates first mediated impact in neurons to a substantial extent. And we are pursuing these mechanisms for therapeutic aspects. So what exactly these proteins do? It looks like, because both RNA binding proteins are involved in the final step of DNA break sealing, it looks like these proteins act as a glue or some kind of stabilizer. When there is a break, ligase has to seal it, but before that, it will be breathing, when the DNA is broken. So it's important to put them in the right place. And that is exactly what is done by first and TDP43. And that's our hypothesis. Like as it does in stress granules via phase suppression, it probably plays a similar role in DNA damage sites where it, kind of acts as a glue for a dynamic protein-protein interaction complex to stabilize the broken ends. So the final paper I want to mention, this is extremely important as a proof of concept. So in this paper, we showed that during brain hemorrhage, when there is a blood, hemoglobin breaks down, the hemin is the first product, and then hemin is further degraded by heme oxygenase to biliverdin and iron, and finally carbon monoxide. So iron has to be taken care of. So it is a multiple steps of detoxification process. The immediate treatment for stroke, as you know, is removing the inflammation as well as blood clotting. But studies show that frequent microbleeds or frequent injury or impact injury leads to some kind of predisposition to neurological disorders. We wanted to understand whether there is any mechanism that contributes to it. We found that hemin induces DNA double-strand breaks. That's extremely surprising. Most of people thought that it will be oxidative and it induces some ROS and indirectly induced DNA damage. We found that it acts as a radiomimetic, like bleomycin, which is used in chemotherapy. Hemin actually, when gets oxidized, forms a dimer, and it sits in the major groove of the DNA and nicks on both sides. So it actually creates double strand breaks. And the level of double strand breaks is as high as 2 gray of radiation for a 5 micromolar hemin. And the immediate effect is, as radiation induces cellular senescence. We found that these double strand breaks induce a senescence-like state in neurons and neural stem cells. It induces actual senescence. So that's extremely important. So we were initially thinking this is a pathological phenomenon, and even other people, as endpoint, they thought this senescence is pathological, and you have to reverse it. But we found that this is a clear adaptation for the cell. This is extremely interesting. So because once Hamin induces double strand breaks, that probably contributes to neuronal inflammation and senescence. But Hamin, if it's degraded by hemooxygenase into iron, that acute iron increase kills cells by ferroptosis in healthy cells. But if the heme degradation occurs in senescence-like state where you have suppressed transcription, the iron is no longer toxic. So cell kind of buys time to safely detoxify hemin and release iron, and then iron will be sequestered by ferritin and so on. So we show that if you prevent senescence in the early stage, actually the cell death is much more because of iron toxicity. So this is a clear adaptation. That's the reason HO1 is induced. Otherwise, there could have been a constitutive HO1 expression. So to overcome this, we found that as an antioxidant, strong antioxidant, using pegylated carbon nanoparticle in collaboration with Tom Kent in Baylor and James Tour in Rice University, we added an iron chelator to it. And when we use that combinatorial therapy, it not only suppresses senescence, but also chelates iron and prevents ferroptosis. And that, we found that that's very effective in mouse models and culture cells, and we are taking it further for therapeutic aspect. This is important because many times we look at the endpoint pathology, but we looked at more biochemical mechanisms happening and modified the therapeutic strategy slightly, and that was very effective in the long term prevention of neurological disorders. So finally, the overall goal in our lab is to understand how various etiological factors in neurodegenerative diseases leads to imbalance in DNA damage and repair, and how it kind of there's a dynamics playing around here with increased genome damage to inflammation and senescence, and how this dynamics contributes to neuronal degeneration. And although we may not be able to go back and identify what caused the initial disease, because that could be 20, 30 years before the symptoms appear, we think that if we understand the mechanisms of how DNA damage contributes to neuronal cell death, we can intervene here and significantly slow down neurodegeneration, especially in age-associated conditions. So overall, take-home message is specific DNA break sealing defects in motor neuron diseases is caused by TDP43 and I feel this is my last slide. So thank you very much for listening, and I will be happy to take any questions.